Hello, everyone. I'm Eva Harris, and I'm here to introduce our next two fabulous speakers. This is my time to rib Joe, so I'm really excited about it. No, this is the time I get to find out all of the dirt about my friends. So Joseph, is a professor and vice chairman of the Department of Biochemistry and Biophysics at UCSF, and he has a joint appointment at the California Institute for Quantitative Biosciences. And Joe is a man of many hats, which is probably why we get along so well. And you might know him of the fame and fortune of the virus hunting that he does using the virochip that he developed together with Don Ganem, which has been very useful for discovering new viruses like SARS, for instance, as well as characterizing populations of known viruses. And so there's a long and torrid history in this area. And then he has another hat, which is as a serious malaria biologist, and he's going to talk today about that part of his work. It combines a whole plethora of disciplines, including genomics, bioinformatics, biochemistry, and bioengineering, to understand the activity of genes, and which you're going to hear at this point, as well as identifying novel drug targets and new tools for understanding the etiology of infections. And so, actually, I had the pleasure of we were kind of the tag team at the latest round of Howard Hughes' holiday lectures, which is really fun, and it was about essentially infectious diseases as portrayed to the upcoming generation of high school and other scientists. And it was really fun, and it was televised, so I can tell you that, not only does Joe look great with makeup, but he's actually a terrific show person, just showman. And so take it away, Joe.
Hey. First of all, I'd like to thank the organizers, Dan, and so on, to having me here. It's a great pleasure. I want to mention that for that lecture, Eva does a great dance on stage in the lecture. You can see it streaming live, or not live, but streaming taped at the Howard Hughes Medical Institute site, so I encourage you to check out Eva's dance skills on stage. Did I get you back? All right, great. All right. So half my lab does virology, and the other half does malaria biology and drug development. And today's talk is really going to center on basic science. I know that many of you, or some of you, might have hoped that we'd be talking about some kind of four-dimensional microarray, global profiling, proteomics thing. This is actually going to be an old-fashioned basic science talk. And it turned out to be really cool. And first of all, the issue in malaria is probably obvious to a lot of you. It's one of the notorious killers of children. It is one of the most heavy burdens on humanity. The proportion of 2 to 10-year-olds infected with Plasmodium Falciparum, one of the 5 malarias that can infect humans, is shown here. And so you can see where the major part of the burden is in Africa. The global eradication efforts, as you may be aware, are starting up again. They face significant challenges, both from a therapeutic standpoint, prevention standpoint in terms of the vector, the mosquito, as well as development of a vaccine. And you're going to be hearing about in the next talk some innovative vaccine development efforts. It affects somewhere between, literally hundreds of millions of infections, but somewhere around 1 million deaths. And it disproportionately affects pregnant women and children under the age of 5, some of the most vulnerable populations in the world. We'll be talking about Plasmodium falciparum today, the most deadly form of human malaria. The life cycle of Plasmodium falciparum is somewhat baroque. It begins with the bite of an Anopheles mosquito, upon which sporozoites are released into the bloodstream. These sporozoites will eventually end up in the liver, or at least some of them will. There they'll invade hepatocytes and undergo multiple rounds of replication to produce a form called merozoites that will burst forth and then infect mature erythrocytes. They'll continue this cycle of growing inside mature erythrocytes, undergoing multiple rounds of replication, and then bursting forth and repeating the process over and over again. At some frequency, a sexual form of the parasites is thrown off. These are so-called gametocytes in both male and female form. These can be taken up in the blood meal of a mosquito, and the process can begin again. And so a lot of the development of therapeutics and vaccines and so on is focused on this stage or on the prevention of infection of sporozoites. And what I'll be telling you most about today is this blood phase, this so-called asexual erythrocytic cycle. If you've been in the lab and you've stared at mosquitoes through the microscope, you're going to find the next bit a little bit boring. But for those of you who haven't, this is just a little movie I took through the microscope. And this is, sorry for it's a little bit out of focus. These are erythrocytes that are infected with Plasmodium. This one right here is going to burst, and you'll see the merozoites come out. There it goes. And you can see the infection of another erythrocyte right there as this merozoite wiggles its way into the unsuspecting erythrocyte. Now it's in and bouncing around freely on the inside. And if we let this movie continue to run, you'll just see basically all these cells sort of blow up. Here's another one. And you can see another invasion event happening here where actually 2 parasites get in that cell. And if you watch it long enough, there's just no cells left at the end. And of course, it releases lots of junk into the media as well. And you can imagine this in a human infection, especially if it's somewhat synchronized, that this could cause a tremendous amount of problems for the host, and it does. All right, so today's story, though, begins with the electron micrographs that were taken during the 60s. So this is an electron micrograph from Aikawa, who was at Walter Reed during the mid-60s. And Aikawa took a lot of EMs of avian malarias and human malarias and so on. And this is a typical one. This happens to be an avian merozoite. Here's the apical end. Here's the nucleus, mitochondria. And then there's this thing right here, which on this micrograph is labeled SB for spherical body. It's a pretty good label as any. I thought, well, maybe that's some sort of storage system or storage place for the mitochondria. Wasn't really clear at the time what that might be, but it was noticed in a lot of plasmodiums that you could see these things. We now know that that storage body or spherical body is what we call the apicoplast today. It's a distinct 4-membrane-bound organelle that's likely of secondary endosymbiotic origin. What I mean by that is somewhere in ancient history there was a primitive cyanobacteria that got gobbled up by a eukaryote. That became some sort of red algae. And then another eukaryotic host gobbled it. So there's a gobbling and then another gobbling. And that's how you end up with 4 membranes, the inner 2 being the inner outer of the gram-negative ancestral bacteria, and then the other 2 coming from the second host, or the first eukaryotic host, and then the second eukaryotic host. And most people observe 4 membranes. There's little controversy about this now. And it's assumed that a red algae is indeed the ancestral guy before Plasmodium. Now, this whole field of the apicoplast in malaria got very heated and excited in the late 90s or mid-90s, around 1996, when Williamson sequenced the genome of the apicoplast. It has its own genome. I'm showing a diagram here. And Jeff McFadden localized the genome to the organelle. So we knew that this genome belonged to that. It wasn't something in the mitochondria or something else. This has a 35 kb genome. It has its own RNA polymerase, RPOB style prokaryotic polymerase. It has its own superoperon of translational machinery, tRNAs. It contains some import machinery and some of its own chaperones. That'll become somewhat important a little later on. And so this was very exciting, of course, because this might be the so-called Achilles heel of malaria. A prokaryotic piece inside a eukaryotic organism should be targetable by drugs that wouldn't affect the host, because you are also a eukaryote. But drugs, some of the antibiotics, and it was already known after this was discovered, it was always known prior to this, the antibiotics had some peculiar features in malaria. I'll show you one here from a paper by Ramya et al. This is clindamycin treatment, and you're looking at relative % survival of the parasites with time. The asexual cycle is 48 hours. A typical drug assay will count cells somewhere around 72 hours and look for % survival. If you use a drug, an antibiotic that affects replication, transcription, or translation in a prokaryotic cell, this kind of effect is often seen, where if you count at 48 hours, it doesn't look like the drug's doing much, not that effective. At 96 hours, it's much more effective, this so-called delayed death phenomenon. And this is why, for acute treatment of malaria, these kinds of antibiotics are not the best. They're often used as a prophylactic or in combination with something else. And so I'm showing clindamycin here, but whether you use erythromycin or tetracycline or rifampicin, very similar effects. In 2005, my lab, in collaboration with Phil Rosenthal's lab at SF General, did a little transcriptomic profiling, and that's the only profiling you're going to see in this whole talk, actually, in which we actually looked at the transcriptome of malaria, and it is this nice, beautiful, single cascade of gene activation that begins and proceeds through the 48 hours and then repeats itself. And we look at untreated and treated parasites, here using 1 micromolar of doxycycline. We were a little depressed at first to realize that there were no credible changes in the entire nuclear transcriptome whatsoever, even though these parasites are guaranteed to die shortly thereafter. Which was a little different than yeast and other organisms in which they're constantly sensing their environment and having a dynamical response to the changing stresses that they're encountering. Not so much in malaria. It was almost like the late lytic cycle of a large herpes virus, in which there's a set program, and once the train leaves the station, there's no turning back. However, there were transcriptional changes in the apicoplast, not shown on this diagram, it's not part of the nuclear genome. And if you look, when it was all said and done, what was realized was that the apicoplast genome was being failed to be replicated and failed to be transmitted to the daughter progeny. So the new merozoites don't have apicoplast. So when they get into a new cell, then they die, which is why we believe that this accounts for the delayed death phenomena. So it's merely a failure to replicate the apicoplast. It also implies that whatever products the apicoplasts are providing to the cell, at least in that first cycle, are already there by the time the defect in the apicoplast is relevant. It was also known from the work of John Boothroyd and others that RNA mutations in plastids, so toxoplasma, for example, has an apicoplast. In fact, I should mention, almost all apicomplexan have plastids or apicoplasts. There's only a few minor exceptions. It was known from the work of Boothroyd and others that clindamycin mutations that made them resistant occur in the apicoplast. So this perhaps wasn't that revolutionary an idea that the mutations would be here, and this was the reason for delayed death. All right, so one of the things that folks started to do soon after mapping out the apicoplast and realizing what a great drug target it might be was to map metabolic pathways to it. So this is a diagram taken from Ralph and from 2004. And people could, by virtue of the fact that many of the proteins that are targeted to the apicoplast contain signal sequences in transit peptides, try to predict what might be in there. And here are the major things that people determined, that many researchers determined were likely to be there. First of all, there's all the maintenance stuff, which we talked about a little bit. There's type 2 fatty acid synthesis, which is very similar to prokaryotes, and I'll tell you about that in a minute. And then there's isoprene subunit synthesis, which we'll talk about a lot, and then heme biosynthesis, which is a funny cycle in malaria because it looks like part of it occurs in the mitochondria, part of it might occur in the apicoplast, and so on. All right, so we already mentioned that, well, I have to go back, step back a minute and just say that although this is what people can map to it, there's a good, solid several hundred genes that are putatively targeted to the apicoplast, which are completely of unknown function. So while I'm showing you this bare bones diagram, there could be a lot of other things going on in there, which you just don't know because you don't know what the genes do. Now, I mentioned that antibiotics take out all this stuff. So we know what that does. So we just ignore that for a minute. The one pathway that got people initially very excited was this one, type 2 fatty acid synthesis, where in a Nature paper in 2000, it was claimed that triclosan, a common antibacterial, you sometimes find it in soap, whether it should be there or not, and that's another issue, was effective against malaria. Not as a drug, but it just killed malaria in vitro. It's not a very good EC50. And it claimed that the target was type 2 fatty acid synthesis. It was difficult, very difficult to reproduce this work. My lab tried to reproduce it. We got very excited by this result. We could never reproduce it. And in fact, when a genetic knockout was finally accomplished of the key enzyme, the target of triclosan in this pathway, the parasites were fine. It's not the target of triclosan. And that of course, was a big-time bummer for the field, to put a technical term on it. Because a lot of people have been working on it, and so it's really important to think about what's a genetically validated target before you go after it. And that one was not genetically validated. And it burned folks, including me to some extent, but we dropped it like a hot potato after that. All right, so that's not necessary, at least for life in the asexual blood stage of malaria. So we can erase that from the screen. This is about the time in my lab when a new postdoc joined the lab, Ellen Gay. She was also a pathologist at Stanford. She's somewhere here in the audience. And she began looking at these pathways and trying to figure out, well, what are the other outputs of this? And the other obvious output in this pathway, besides heme biosynthesis, is isoprene unit synthesis. Now, Plasmodium uses a bacterial type isoprene unit synthesis pathway, the so-called non-malevolate pathway. It begins with different precursors than the eukaryotic ones, pyruvate and glyceraldehyde 3-phosphate, instead of acetyl-CoA and acetyl-CoA condensation. And so it uses different enzymes, different starting products, yet arrives at exactly the same products, IPP, isopentyl pyrophosphate, and dimethyl allyl pyrophosphate. And these 5-carbon building blocks build all kinds of important things for the cell, various kinds of small molecules, prenylation modifications in proteins, and so on. And so different pathway, same products. So it's still a good target. Now, what was known about this pathway is that there is a drug, in fact, that is known to inhibit one of the enzymes, the one that was starred back there, DOXR, in the pathway, phosphatomycin. It's published that if you treat with phosphatomycin, parasites die. Well, is it just like triclosan, where parasites die, but that's not the target? This is a little different than the type 2 fatty acid pathway in that you can actually, we know what the products of this pathway are. So Ellen reasoned that if we supply the products, and just cover with the metabolite, that we could overcome the treatment with phosphatomycin. And if that worked, that would in fact validate that pathway as the true target of that drug. And then it opens up some doors. That is, you could label those subunits and go chasing them and so on. We'll talk more about that in a minute. So let's talk about the relevant experiment here. The first thing she did is try DMAP to rescue those. And I'm not going to show you the data, because nothing happened. It didn't rescue at all. So total failure. But something different happened with isopentyl pyrophosphate, IPP. So here's the kill curve for phosphatomycin, percent survival, and so you get a nice kill curve with the drug. At 25 micromolar IPP, it pushed the curve a little to the right. At 50 micromolar, the curve continues to march to the right, indicate there really is effect here. At 100 micromolar, now you're beginning to achieve rescue. At 200 micromolar, this is definitely what I would call rescue. At 400 micromolar IPP, now the drug has no effect at any of the tested concentrations. So 500 micromolar IPP produced a complete rescue as phosphatomycin-treated parasites, essentially all concentrations tested here. So we can conclude that IPP is the essential byproduct of this pathway, and phosphatomycin really does target that pathway, which is important. Now, recall the antibiotic treatment. The antibiotic treatment takes out replication, translation, or transcription from this side over here. And so if we remove that, we can ask the very simple question. Would supplementation of IPP also rescue the complete loss of the organelle induced by antibiotic treatment? Essentially asking the question, is the only thing this complicated organelle does, in the blood phase at least, is produce a single essential 5-carbon molecule? You know, that's not intelligent design, if it does. OK, so here is the experiment. And obviously, I wouldn't be telling you about it if it didn't turn out to be awfully interesting. Here's doxycycline. You get the typical delayed death phenomenon. Instead of at 48 hours, you see most of the death past 96 hours. When Ellen added IPP to the media, she obtained a rescue, which is shocking. And then when you withdraw the IPP, the parasites die. They're dependent on it. OK, now, I'm going to argue here that what has occurred is that we have removed the organelle completely, and they don't need it, that this is the only thing. You can't argue with the conclusion that IPP will rescue the treatment from antibiotics. But let's go a little further and try to establish if the apicoplast is truly gone or not. The first thing we can do is measure if the genome is there. And so just by qPCR asking, is the apicoplast genome around anymore? Is it being lost like we saw previously in previous publications where you treat with antibiotics and you southern blot for the genome and it disappears? So by qPCR, sure enough, we see complete loss of the genome to undetectable levels after only about 200 hours. And we've carried out parasites now for weeks and weeks and weeks on IPP, and there is no detectable genome there. All right, that's just one measure. In yeast, you can lose the mitochondrial genome, but you can still have the mitochondria around. So rho minus yeast. So is it something like that? What's going on with proteins that are targeted to the apicoplast? Because maybe it's still just doing its thing, but at some reduced level or something like that. Well, some transport of proteins to the apicoplast is guided by a signal sequence and a transit peptide. This is cleaved off in the ER, and this is cleaved off upon entry into the apicoplast. Jeff McFadden made a really great construct some years ago in which they fused GFP to the signal peptide and the transit peptide, allowing them to track GFP being targeted to that organelle. And this is how it appears from one of their publications, just this very discrete dot. And so we can actually monitor that, first initially just by Western blot, to see if the proteins are in fact processed as one would predict. So the very simple prediction is if we treat with IPP and antibiotic, all the proteins won't be processed. If there's still organelles around that are capable of processing and folding these proteins correctly, they will get processed. Here's untreated parasites, unprocessed, processed, and this is this construct here. You can't, this guy gets cleaved off too fast to see in the ER, so we're only looking at this cleavage right here. In doxycycline-treated parasites, what is observed here is the typical processed band, and then right before they die at 96 hours, you see a shift to the unprocessed, indicating essentially elimination of the apicoplast, and then, of course, there are no parasites to assay. If we co-supplement now with IPP and doxycycline, we see a shift to the unprocessed band. There's some degraded stuff that we do see in some of the other photos, but basically, all the protein is now in the unprocessed fraction, and we see no evidence of processing occurring. Which our interpretation of this is that there's not a functional organelle around to cleave the transit peptide, and therefore the machinery is not there to fold these proteins, and therefore the compartments are not there to carry out any of the functions that would be carried out normally if the genome was present and the organelle was there. Okay, we can look at this by microscopy as well and ask a couple of questions. This is where the GFP apicoplast construct goes normally. It's a dot in the beginning, ring stage. Trophozoite stage, it's just a bigger dot. And after replication in Schizonts, you see the apicoplast now segregated to the daughter merozoites prior to release and invasion of new cells. If we do the doxycycline and IPP rescue, we get this. So it's very diffuse early on. It's more diffuse. And later on, it just looks kind of like just diffuse punctate stuff, like a little grenade went off inside the cell or something. It's not localized very well. This is some evidence. It looks somewhat fuzzy. It's hard to see what's going on here. So we use the new Nikon A1 confocal microscope in the Nikon Imaging Center at UCSF, a microscope, by the way, which I cannot recommend more highly, to do some confocal 3D reconstructions. I don't own any Nikon stock or have any financial involvement whatsoever in Nikon, but I can say I do like the pictures. This is DAPI staining, so this is the nucleus. Here's the GFP signal. Here's one. We could show you many parasites, but they basically look like this. These are normal parasites, so you get discrete little organelles. In the apicoplast apicoplast minus rescued parasites, this GFP is just all over the place. There's no one discrete thing. Okay, so but what is that punctate stuff that's floating around? Could that just be little pieces of apicoplast that really are just functional and it's carrying out the function, but they're not forming just one cohesive organelle anymore? Maybe that's part of it. So and everything I've showed you up to now, by the way, is published in PLOS Biology just recently. What I'm going to show you now is now transitioning to unpublished data. So to do that, to look at these GFP signals a little more closely, we resorted to EM and immuno-EM. And so here's a normal erythrocyte by electron microscopy, and we see the nucleus here and the food vacuole here. Here's the erythrocyte, and here's the parasite itself. The food vacuole is always easy to see in parasites because of these massive hemozoan crystals that they contain. It's very diagnostic. If we use the GFP targeted strain and immunolocalize that GFP, we can see where the apicoplast is. So here is immunolocalized GFP. It's the nucleus out here, presumably. And this is what we believe to be the apicoplast. Here's a gratuitous zoom. And to show you that you can actually see the four membranes of the organelle. And that is, of course, one of the hallmarks of the apicoplast, because it's a four membrane-bound compartment. So we're convinced that's the apicoplast. That's great. What about so-called apicoplast minus rescued parasites? So here's without immuno-EM. They look kind of like normal parasites. They don't look that much different, with the exception that you see these little dense vesicle-like looking things floating around in the cytoplasm all over the place. And in many, many cells, we observe this. So I can zoom in on those, and you can see these guys. You don't see these in the wild-type parasites. You see these in the rescued parasites. Immuno-EM can come to the rescue and see if they're GFP positive. So here's an immuno-EM, and we can see localization to what look like small vesicles. And if we zoom in there, they look like single membrane-bound vesicles. Our interpretation of this is that these are transport vesicles from the ER to nowhere, because the apicoplast is gone. And so this is cargo intended for the organelle, but they've got nowhere to go. And because the nuclear genome doesn't care if the apicoplast is gone or not, and it really is not dynamically sensing the presence of the organelle whatsoever, it just keeps cranking these things out as if it's a happy day. But it's not, at least for the parasite. All right. And so these things are just floating around, and we think this is an accumulation of apicoplast intended cargo with nowhere to go. We could show you more of these kind of things. Here's another electron micrograph of the same thing. If we zoom in, we see an accumulation of these little vesicles, and we see also that they're GFP positive. All right, I want to make one conclusion here before we move on to another part of the future directions and what else, some more recent data. So the overall conclusion I can make from this right now is that the only essential function of this entire organelle during the blood stage of growth is solely the production of isopentyl pyrophosphate, and that's it. The other stages I can't speak to yet. I'll show you a little preliminary data in the other stages. So that gets to this. We're just talking about this. What about the next stage, which is the sexual phase development? At some frequency, these parasites can develop into male and female-formed gametocytes. Is the apicoplast necessary for this development? So Vita Ion in the lab conducted an experiment where she induced gametocytogenesis, which you can do just by sort of stressing them out a little bit. And lo and behold, these gametocytes develop just fine. And even a little more surprising is that not only do they develop just fine when you add IPP in parasites that are already made to be apicoplast minus, but after they start to develop, if you remove IPP, they don't care, and they continue to develop along the way, all the way to stage 5, which is the last stage before they should be taken up into a blood meal, in the absence of IPP, implying that there is no product of the apicoplast that's at least required for these developmental steps. However, a rightful criticism of this is you don't really know that these gametocytes are competent for mating and making the next stage, which would be an oocyst in a mosquito. So after the gametocytes are taken up by a mosquito, this is where mating occurs. They're haploid all through this part. They're transiently diploid in the mosquito, and there's a complex developmental cycle that occurs there, which I'm sure you're going to hear about more in the next talk to some extent. And so together with Shirley Luckhart at UC Davis, who's formerly at Walter Reed, we've taken a very preliminary experiment by feeding these gametocytes to mosquitoes and asking, do they make oocysts? This is the form preceding sporozoite development, where it's a developmental form that's basically crossing the midgut wall before they release sporozoites that will then travel to the salivary glands. And at least in our preliminary experiments, they're very encouraging in that apicoplast minus parasites do in fact make oocysts. We can at least say that. We don't know if they're at a disadvantage or they're making less of that. The N here is just way too small. This is a pilot experiment and should be taken with a big fat grain of salt. It's got to be done over and over again. And we've got to do it with now sufficient numbers that we can do some counting and so on. And of course, a real key question in here is, after this stage, will they make sporozoites? And that'll be relevant for something I'll talk about right at the end. All right, so I want to make a couple points here. Apicoplast minus parasites enable actually a wide variety of experiments. This is a new tool that hasn't been available before. So because we can take the apicoplast out, we can ask questions about these different things. So for example, if we have no apicoplast, these GFP vesicles, or the cargo vesicles intended for delivery to the organelle, accumulate now at a decent rate. These, by virtue of the fact that they're labeled in this case with GFP, can be purified out by a variety of different biochemical methods. And then we can go to town in mass spec land and proteomically characterize the intended cargo for the apicoplast, which would then allow us to compare those to, say, bioinformatic predictions of what we think might go to the apicoplast. That's one thing that we can do. The other thing we haven't answered here very clearly is, we know IPP is required, but what exactly is IPP doing for the parasite that's essential? We haven't answered that at all with these experiments thus far. But by virtue of the fact that all their IPP must now be exogenously supplied, we can either come in with labeled IPP or some affinity reagents. So for example, carbon-13 IPP, and chase those things down in a sort of traditional biochemistry fashion. Are there critical or essential prenylations? What small molecules might they be making? Isoprenoids create some of the most diverse small molecules that are ever known. In fact, artemisinin, a leading drug against malaria, is made from isoprenoids, though clearly not in malaria. So this allows us to chase those down. I mentioned that there. The other thing I want to mention here is that by covering parasites with IPP, one can conduct a forward genetic screen. And that's not been possible in malaria before. For a genetic screen in malaria, you don't really hear that sentence that often, just because it would be so much of a pain. But here you can actually do it. You can actually knock genes out under cover of IPP, then withdraw IPP to know if those genes are essential for production or maintenance of the apicoplast. And that hasn't been possible before. And so that's an exciting new area of development as well. And finally, there's drug screening. There are lots of drugs. You know, one of the dirty secrets of malaria drug screening is there's a lot of bioactive compounds against malaria, whether or not they're good drugs in humans or not. There are a lot of bioactive compounds that are potent against malaria, but the targets are completely unknown. I was just talking with Kip Guy at St. Jude yesterday. He's writing a review on this subject, and he estimates that around 80% of bioactives against malaria have no known target. And he's collated data from Novartis and GSK and so on. And so some of those might target pathways involved in the maintenance or production of the apicoplast itself. And you could, for once, figure that out. You could take bioactives and then, of course, do a parallel screen with and without IPP. And those that rescued in IPP, you automatically know that that drug, in fact, targets something to do with maintenance or of the apicoplast or its critical pathway, generation of IPP. And so that's an exciting pathway-specific drug screen. I've mentioned these kinds of experiments. I'm going to end with one other possible kind of use for this. And I know this gets into a more sort of fantasy talk, and you're going to hear a lot about more realistic talk about vaccines in the next one. But this is an interesting candidate, possibly, for an attenuated vaccine. Because they depend on IPP, and there's no detectable IPP in the blood of people, it's an incredibly membrane impermeable compound. Note that we had to treat these guys with 400 micromolar IPP. And they are, infected erythrocytes are permeable in ways that uninfected are not. You could theoretically give patients, let's just do the easy, kind of the stupid way first, give patients infected erythrocytes. There's problems with that, obviously, but give them infected erythrocytes. Those parasites are guaranteed to die in the next cycle. There's no IPP. They permanently lost their apicoplast, and there's no way for that to revert. You can't get those genes back. And so at least for one cycle, you would have the benefit, the host would have the benefit of seeing a near wild-type parasite, which does antigenic variation. The innate and adaptive immune responses would get a little bit of benefit from seeing that. And potentially, through repeat infections, you could confer some sort of limited immunity. That would be the hope. Now, I showed you the sporozoid development. That would be the real fantasy. We have no idea if these things are going to develop past oocysts. But if, in fact, Apicoplasmitis parasites were able to develop into sporozoites, now you have a transmission vector that you could actually administer to patients and perhaps give you, sporozoite-level vaccine or protection. And then it could even be backed up at the blood stage as well. And there's novel strategies for that you're going to hear in the next talk. So that's the thought I want to leave you. I know I've gone a little faster than the typical talk, but that'll leave more room for Steve, right? And so I'm going to end there. Ellen Ye did most of the work. This is the rest of the lab that also supports Ellen, and a lot of the malaria and virus people are also shown on this slide. And so with that, I'll take some questions.
You alluded to this, but does it seem reasonable to you that that complex organelle would have the sole function of it would be to make a single organic molecule or two?
I'll try and repeat the question. Are you asking whether I'm surprised by the fact that this appears to be the only function? Yeah, that's the question? Yeah, I'm surprised. So in fact, when Ellen said, let's try and knock out the whole thing with the antibiotics, it's probably not going to work. And I said, hell yeah, it's not going to work. That's a crazy idea. And also, it's crazy because this compound's really expensive, which may be why no one ever did it before. It's sort of interesting. That's another topic. But I am surprised. There's some interesting, in retrospect, maybe it isn't that surprising. So Babesia bovis is another apicomplexan parasite of actually some significant importance. It's an apicomplexan parasite of cows, obviously, and it is very similar to malaria in the fact that it infects erythrocytes and so on. And Babesia bovis, it has already been published actually prior to our work on this, that it's, as an organism, it has completely lost the type 2 fatty acid pathway. It's gone. It doesn't exist. It also lost the enzymes for de novo heme biosynthesis. I didn't mention it, but our results directly imply that de novo heme biosynthesis is not required or essential in the blood phase of malaria, which is that I don't find surprising. I mean, for goodness sakes, they're drowning in heme, man. But Babesia bovis lost type 2 fatty acid synthesis, lost de novo heme biosynthesis, and what remains only in the apicoplast function there is synthesis of IPP. And so the writing might have been on the wall on another apicomplexan. I don't know who to call, or someone direct. Am I supposed to point them out? Yeah. All right, you.
Hello. So if you culture for a long period of time just with IPP, do they kick it out?
Do they keep going?
So yeah, if you kind of supplement with IP for a significant period of time, will they kick it out?
Will they kick it out?
Well, will they lose the apicoplast? Without having the pressure?
Without pressure from the antibiotic. That's an interesting question. We haven't done that experiment. I can imagine. I'll just grab that from you. You can put that right there. There you go. There you go. I can imagine that. Well, actually, I don't have a good prediction what's going to happen in that experiment. That's an interesting experiment, and we haven't done it.
Because that's going to be, just for the kind of forward genetics, it takes a long time.
Yeah, exactly. And did I mention IPP was expensive? So if you have to culture for a long, long time just to get that answer, I definitely encourage someone to try it. Is there another question back there that I'm missing? No?
Oh, hello. So from your talk, so you have looked at apicoplast in Plasmodium Falciparum. So is apicoplast also present in other strains of malaria?
Yes. So.
And do they function the same?
Well, that's an interesting question. So first of all, almost all apicomplexans, which are all apicomplexans are parasites. Almost all apicomplexans, with relatively few exceptions, have apicoplasts, organelles, and plastid genomes. Is the function the same? So in Babesia bovis, maybe it's very, very similar to Falciparum, and it's just a more advanced stage of losing those genes. Toxoplasma, very different organism, very different physiological niche. And when the organism is in a very different physiological niche, I don't think we can make the same assumptions about what is required. So that's the answer. All righty, that was fun.